Dear colleagues, I am Dr. Serol Korkmaz from the Veterinary Faculty of Cukurova University. Today, I will present our research results which were published in the journal MVZ Cordoba, Colombia. This article examines the effects of myo-inositol supplementation on the nutritional contents and physicochemical quality and hygiene of wet and dry cat foods. Feed hygiene is crucial for both pet and human health, as poor practices can lead to contamination by pathogens like viruses, bacteria, and fungi. Contamination can happen during various stages of feed handling, processing, transport, storage, and contributes to feed-borne disease risks and zoonotic transmission. Virus survival in feed is influenced by the feed's nutritional composition and environmental conditions such as temperature, humidity, pH, and storage. Ensuring feed hygiene and safety through proper monitoring of production and storage is crucial to prevent contamination and spoilage. To support this, various feed additives such as preservatives, antioxidants, stabilizers, and acidifiers are used to inhibit pathogen growth and extend shelf life of animal feed. Ensuring feed hygiene and safety through proper monitoring of production and storage is crucial to prevent contamination and spoilage. To support this, various feed additives such as preservatives, antioxidants, stabilizers, and acidifiers are used to inhibit pathogen growth and extend shelf life of animal feed. Nowadays, natural polysaccharides from plants, algae, and microbes have emerged as effective preservatives with antimicrobial activity against viruses. Myo-inositol, a natural compound found in animal and plant tissues, plays a structural role in membranes and is approved as a nutritional additive for pets and fish. For this purpose, the study investigated the effects of dietary myo-inositol supplementation on nutritional ingredients and physicochemical parameters in feed structure such as pH, water absorption index, and the water solubility index of dry and wet cat foods. Additionally, the interaction of these physicochemical parameters and the viability of murine norovirus as a surrogate of feline calicivirus virus were investigated, and the half-life of the virus was determined as a function of interaction between storage time and temperature in dry and wet cat foods for feed hygiene and safety. The powder form myo-inositol was supplemented at the 0, 12.5, 25, and 50 milligrams in 100 g wet and dry cat food. Crude protein, CP, crude fat, CF, crude fiber, moisture, and ash contents of the samples were analyzed in accordance with AOAC methods. Then, 1 gram samples of dried feed in a centrifuge tube at room temperature or in a refrigerator condition for 30, 60, and 120 minutes. For simulation viral contamination, the virus suspension was sprayed onto 100 grams of feed using a plastic sprayer inside a BSL-2 cabinet. Feed samples with the virus was stored at room temperature or the refrigerator during 30, 60, or 120 minutes. Myo-inositol supplementation, storage temperature and storage time did not cause any difference in proximate parameters of dry and wet food. For both food type, myo-inositol supplementation dose-dependently decreased the viability of virus in all storage conditions of foods. Similarly, the virus titer was decreased by increasing the storage temperature and time with a weak correlation. In wet cat food, the virus was completely inactive after the storage time of 120 minutes at room temperature. However, any significant correlation was not determined between nutritional contents, physicochemical measurements of both food types, and the virus viability. In conclusion, myo-inositol concentration, storage time and temperature were ineffective on pet food nutritional and physicochemical properties, but they were effective on viral load of pet food